It is a pleasure to be here to share with you some of the work that we're doing at the Schoen Adams Research Institute, which is a part of Spaulding Rehabilitation Hospital. Exercise is medicine. 3 words I imagine resonate with most of the individuals in this audience. And that is because most, if not all of us, can readily recognize and appreciate the widespread beneficial effects of exercise for overall health and well-being. Exercise is important for the maintenance of health, and it's also important for the recovery of health and function after injury or in the setting of disease, as is the case with rehabilitation protocols. And exercise is also a potent antidote to counteract many of the negative effects of time's arrow on cell, tissue, and organ function. For a multitude of hallmarks of aging, or what can be considered common denominators of age-related cellular declines, it has been shown that exercise exerts potent rejuvenating effects. And these rejuvenating effects could not come at a more important time, where our aging population continues to expand at a remarkable rate. It has been estimated that within the next 2 decades, over 90 million individuals in the US alone will be age 65 and older. Now, this is really an exciting testament to medical advances. Medical advances, as we've already heard this morning, and as we'll be continuing to hear over the next 3 days. But with these exciting new medical advances and the enhanced lifespan that they afford, so comes a need to develop technologies that will concomitantly enhance health span. For we know that as our population gets older, so does the presentation of numerous physical and cognitive declines. In our laboratory, we are particularly interested in the so-called muscle-brain axis, whereby it has been shown that even short-duration, low-intensity exercise can exert significant beneficial effects on cognitive function in older individuals. However, the mechanisms underlying these benefits have been largely unknown and poorly understood. We can get some really interesting clues, however, from some recent studies that have demonstrated the role of circulating factors in dictating aging phenotypes. Investigators at Stanford University have used this experimental paradigm called heterochronic parabiosis, whereby a young animal is joined together with an aged animal counterpart, such that their circulatory systems are exchanged. These studies have demonstrated that age factors from the older animal can induce an accelerating aging phenotype in the young animal counterpart. In contrast, and perhaps more importantly, it's been demonstrated that young circulating factors can induce rejuvenating effects in numerous aged tissues and organs, including the brain. So we and others have been very interested to better understand what might be those critical factors within the young blood that are responsible for those beneficial effects. Recent studies have demonstrated that a large portion of the circulating secretome is packaged within membranous nanoparticles called extracellular vesicles. Extracellular vesicles are released by an originating cell into fluids throughout the body, including the blood. These extracellular vesicles then traffic between anatomically remote sites where they serve as critical couriers of information exchange. Importantly, through this information exchange, extracellular vesicles can target and reprogram recipient cells, thereby modulating physiological functions and pathophysiological cascades. From a therapeutic perspective, extracellular vesicles are particularly advantageous because they are immune privileged, they are biocompatible, and they also offer the advantage of readily crossing the blood-brain barrier. So as you can see here, we performed some studies where we fluorescently labeled extracellular vesicles and we intravenously injected them into an aged animal. Within 24 hours, we saw an abundance of our donor-derived extracellular vesicles throughout the brain. And this really represents an important advantage for extracellular vesicles, since a major limitation in the development of therapeutics for CNS applications has been the fact that only approximately 1% of proteins are capable of crossing the blood-brain barrier. So as a next step, we wanted to expand on some of our previous work demonstrating the ability of young-derived extracellular vesicles to significantly enhance muscle stem cell function and age skeletal muscle regenerative capacity. So we decided to extend our studies into an aging brain model. So this is just an overview of our experimental paradigm. We randomized aged animals to receive serial intravenous injections, and we evaluated outcomes after several weeks. Specifically, we looked at 2 cognitive outcomes: novel object recognition, which is a metric of recognition memory, as well as fear conditioning, which is a metric of associative learning and memory. When we did this, we started off just comparing 2 groups: aged animals that received young serum intravenous injections versus animals that received saline injection controls. And here what we saw, consistent with previous work and previous published manuscripts, was that administration of young serum significantly enhanced both learning and memory in our aged animals when compared to saline injected controls. Next, we wanted to look at the direct effect of extracellular vesicles to evaluate whether they may be potentially mediating some of this beneficial effect that was observed. And so in a third group of aged animals, what we did is we again performed a serial injection paradigm of administering young blood, but this time we depleted the young blood of all extracellular vesicles. And here what we found was that when we removed the extracellular vesicles from the young blood, well, then that beneficial effect on cognitive outcomes was significantly blunted. And in fact, there was no difference between those animals receiving extracellular vesicle depleted serum versus those that received saline injection controls. So as a next step then, we're very interested in honing in on the key biomolecular cargoes within the extracellular vesicles that may be responsible for these effects. And our idea is to engineer extracellular vesicles to enhance this critical payload. The biomolecular cargoes within extracellular vesicles are quite vast. And so to hone down on specific targets, then, we've been leveraging our knowledge of this inverse relationship between aging and exercise. Specifically, we have been employing computational analyses and machine learning algorithms to evaluate those biomolecules that are decreasing with increasing age and changing with exercise. We have already identified several candidates. For example, we currently have a patent pending for one candidate to be engineered within extracellular vesicles. This candidate is the protein Klotho, which has actually been shown to demonstrate important synaptogenic and neurogenic effects in the aging brain, as well as a number of other longevity functions. Another one of the cargoes that we have identified using our computational analyses is currently in preclinical testing, and we are preparing to submit for IP disclosure. There are another 3 candidates that we have identified, which are currently transitioning from computational analyses into experimental designs. We're also very interested in pursuing a compounding approach, whereby we want to test whether administration of 2 or more of these molecular cargoes may exert additive, potentially even synergistic effects. So as a next step then, we want to dig further into evaluating these biomolecular cargoes. We are optimizing cargo engineering within extracellular vesicles. We want to conduct an array of dosing studies, as well as evaluate extracellular vesicle kinetics and biodistribution. For these studies, we anticipate that approximately $2.5 to $3 million will be needed to fund the studies for each cargo. And so we're looking for partners to advance this technology toward IND with the ultimate goal of enhancing the cognitive health span of our aging population. Thank you.